Okay, thank you all for coming tonight. It's my real pleasure to introduce our faculty lecturer, Hugh Rosen, who is a member of the departments of chemical physiology, immunology, and molecular and experimental medicine. He's also a chair of TSRI's Committee on Advanced Human Therapeutics. He was born and raised in Cape Town, South Africa, where he received his bachelor's and MD degrees from the University of Cape Town. He then moved to England, and he obtained a DPhil in physiological sciences from Oxford University. After Oxford, Hugh moved to Merck Research Laboratories in 1990, and over the ensuing 12 years, moved quickly through the ranks to executive director of immunology and rheumatology. In 2002, we were very fortunate to recruit Hugh to TSRI as a member of our faculty. I actually recall this moment quite well because it coincided temporally with Hugh's landmark publication in Science describing the mechanism of action of the immunosuppressive drug FTY-720. And every so often in one's career, you read a paper and you just think, wow, I really wish I had done that. And that was my experience of admiration/envy when I read Hugh's paper. At that time, FTY-720 was a drug under clinical development in Novartis, but the competitor company had essentially no idea how it worked. In an elegant series of studies, Hugh's group showed that FTY-720 acted as a prodrug that undergoes phosphorylation in vivo to become a broad-spectrum sphingosine 1-phosphate receptor agonist, which promotes sequestration of lymphocytes in secondary lymphoid organs to produce the immunosuppressive activity. I found the paper particularly remarkable because it bridged three fields that I would argue have largely operated in isolation from one another, that being immunology, pharmacology, and lipid biology. Using his knowledge of sphingosine 1-phosphate chemistry, and Hugh deduced the structural similarities between the lipid and the FTY-720 drug, and then showed that a stable phosphonate analog of the drug recapitulated its immunosuppressive activity. This research, in my opinion, was chemical biology at its very best, and its impact on both basic and translational sciences is hard to overstate. As most of you, or many of you at least, are probably aware, FTY-720 is now a billion-dollar drug used in the clinic to treat multiple sclerosis. And while Novartis may be benefiting financially from the drug, it was Hugh's group that actually figured out how it worked. Interestingly, Hugh's studies, as well as others, have pointed to potential side effects caused by these sorts of broad-spectrum S1P agonists like FTY-720. And at TSRI, Hugh has set out to address this important challenge. And you'll certainly hear more about the science behind this remarkable story today in Hugh's lecture. So I will only note that an output of his work, performed in close collaboration with Ed Roberts here at Scripps, is an S1P1 selective agonist termed RPC 1063 that is now in phase 3 clinical trials as a safe and efficacious treatment for multiple sclerosis as well as other autoimmune disorders. Before handing the stage over to Hugh, I wanted to briefly comment on his tremendous impact on the larger TSRI ecosystem. Prior to Hugh's arrival at TSRI, our institute had essentially zero capacity to perform high-throughput screening for small molecule active small molecules, which greatly limited our biologists' access to chemical probes, and conversely, our chemists' access to biological assays to test their synthetic small molecules. Hugh, along with our colleagues in Florida, including Pat Griffin, Bill Rausch, among others, have fundamentally changed the culture of TSRI by building, without question, the top academic small molecule screening center in the country. From 2005 to 2014, Hugh served as the PI of TSRI's NIH-supported small molecule screening center, which, beyond bringing in more than $60 million in grant funding, demonstrated, in my opinion, better than any other NIH-supported center, the transformative impact of high-throughput screening and chemical probes on basic and translational biological research. Owing in large part to Hugh's leadership, screening and chemical probes have become terms that permeate the daily lexicon of Scripps research, fostering an environment where frontier pharmacology has taken its rightful place alongside genetics as preferred means to perturb proteins and pathways in complex biological systems. Hugh, we look forward to your lecture today entitled provocatively Of Mechanisms and Molecules from R01 to Intelligent Intervention in MS.
Thank you very much, Ben, for that very generous introduction. When I got the invitation to talk to give the faculty lecture, it was the first week in June, which was, of course, an extraordinarily interesting week here at TSRI. And when I accepted, I said, well, I wonder what TSRI would actually be like as these things evolve through the ringer. And I have to, I think, extend my thanks and admiration to the faculty of TSRI for standing up with its soul and fighting for the heart of our institution, and to Jim Paulson for stepping into the interim leadership as a safe pair of hands. This effort, I think, is very, very much appreciated and is tremendously important for an institution that I have very high confidence will go from strength to strength. And the work that I'm going to talk about today is only one small facet of the many things that are done here at TSRI to push forward fundamental discovery and ultimately improve the life of patients. And one can point to compounds from Jeff Kelly's lab, from Charlie Cochran, some of Barry Sharpless's work that are already fundamentally changing the environment in which we operate. And the outlook, I think, is for a very healthy TSRI that will have many diverse sources of income and be much immune to short-term changes in funding environments. And I think that we just have to keep a steady focus on working towards that, and the outcome will be extraordinarily good. Let me begin by thanking the people that have worked with me on these these particular projects in my lab: Juman Sana, Pedro Gonzalez Cabrera, Wijung Go, Stuart Cahalan as a graduate student, Nora, Sean, and Hallie. In the screening center, Peter Hodder, Steve Brown, MT Schaefer, Lucine, Sean, and Charmaine. Ed Roberts has been a brilliant collaborator in medicinal chemistry, and Miguel Michelangelo, Shime, and Ram have all contributed hugely to the chemistry that was done. We would barely understand some of the mechanisms of immunomodulation without the interaction with Michael Oldstone, John Taharo, and Kevin Walsh. The structure benefited tremendously from an interaction with Ray Stevens and Peter Kuhn. Bill Kiosis has really helped us with our imaging, and then colleagues at Receptos have contributed to the crystal structure. Mike Hansen initially at Scripps and then TSRI. Robert Peach and Marcus with the preclinical development of 1063, and Sheila Gujarati with the clinical development of RPC 1063. And without the NIH, none of this would have started. We stand on the shoulders of giants, and most of us, I suspect, are luckier than smart, and we sort of forget the huge contributions of the intellectual giants who preceded us. And for someone like myself, who's a physician scientist and is interested in therapeutics, the intellectual roots of this were the 19th century notions of William Osler, looking at disease and patients as, in a sense, broken machines. That transition to 20th century medicine by his successor as Regius Chair in Oxford, Archibald Garrett, who described the first inherited metabolic defects and put molecular medicine on a clear scientific and, in fact, chemical plane, using the only tools that he had available, which in those days were chemistry and biochemistry. And then the person in whose shadow I trained at the Dunn School of Pathology in Oxford, Howard Florey, the man who really has always served to me as the fundamental example of somebody who could initiate transformational intervention. And in his discovery and development of penicillin, brought in the antibiotic era and changed the face of medicine essentially overnight. Lewis Thomas, writing in the New England Journal of Medicine in 1974, spoke about intelligent intervention, and he pointed out that the mechanisms of disease are quite open to intelligent intervention and reversal whenever we learn more about how they operate. And this is essentially one of the underlying things that we've tried to do at TSRI. And I would argue that intelligent intervention really reached its high point in some of Ed Roberts' unique insights into medicinal chemistry, because without Ed and this remarkable ability to find the right sweet spot in chemical space across multiple parameters at the same time, I would never have been lucky enough. So we tried to use the power of chemistry and genetics to establish causal relationships between protein expression, signaling, and function in physiology and pathology. What personally I like about chemistry are the advantages of chirality, reversibility, and loss of and restoration of function in real time to establish true causality between a protein and its function and a biological readout. We began with a hypothesis about the way these lipid receptors functioned, used the tools of high-throughput screening and chemistry to make chemical probes that gave new insights into modulating cytokine storm. En passant, allowed the crystal structure of the receptor to be solved, and a compound emerging from this loop ended up in human safety and with demonstrated safety and efficacy in man, and I'll tell you about that in more detail. So this is the first compound actually that is derived from the NIH Compound collection. So the lead was discovered in the NIH Compound collection. The compounds were synthesized in 2008, entered phase 1 in January of 2011, and now is in phase 3 for both relapsing remitting multiple sclerosis, and will make the transition to phase 3 for ulcerative colitis early in the new year. The signaling molecule that I've been focusing on is sphingosine 1-phosphate, and really is a fox and not a hedgehog, in the sense that a hedgehog can only needs to do one thing well, yet a fox does many things. It's a signaling lipid present in micromolar quantities in plasma and around 100 nanomolar in lymph. The vast majority, 99%, is bound to high-density lipoprotein, leaving a free concentration of around 15 nanomolar in blood. These tonic circulating levels that are largely sourced from the synthesis in red blood cells and secretion are punctuated by local modulation. And too much free S1P is fatal. It induces coronary vasospasm, cardiac arrest, bronchial smooth muscle constriction, and pulmonary edema. So we live on a knife edge. The long-chain-based sphingosine has no de novo synthetic pathway. The largest reservoir is in sphingomyelin in the lipid rafts on the external leaflet of the bilayer. This is cleaved by sphingomyelinase to generate ceramide. Ceramide is then N-deacylated by ceramidase to generate sphingosine. The long-chain base is then phosphorylated by 2 kinases in both mouse and man to generate sphingosine 1-phosphate. Sphingosine 1-phosphate then can be secreted to the extracellular environment through the spinster channels. It can also be dephosphorylated back to sphingosine or degraded by a lyase and removed from the signaling pathways. It does, like any fox does, many, many things. And so acting on a variety of receptors has effects on the brain, where it's expressed on astrocytes and neurons, where it also plays a role, as Gerald Chun and colleagues have shown, in hearing and balance. In the lung, it is responsible through S1P3 for generating fibrosis, airways hyperreactivity, and in producing cytokine storm in response to viruses. In lymphoid organs, it regulates lymphocyte recirculation, dendritic cell migration, and the behavior of the marginal zone B cells. In the heart, it regulates rhythm, myocyte survival, as well as vascular resistance, and also seems to regulate endothelial integrity and the leakage within the kidney. So there's almost any system in which one wants to look for a function that sphingosine 1-phosphate acts on this, and it does it through having 5 high-affinity receptors. What we discovered early on in the work on lymphocyte recirculation was that really mid-range S1P1 signaling tone is required for the normal movement of lymphocytes from secondary lymphoid organs back into the blood where they recirculate. So either lymphocyte S1PR1 deletion, as was shown by Rick Proyer and Jason Sister, leads to lymphocyte arrest. Agonist alteration of tone also leads to arrest, and full occupancy of the S1P1 receptor by antagonist also leads to arrest. And in between, there is a titratable range where normal physiology takes place and lymphocytes cross from lymphoid organs across the endothelium into the lymphatic to return to the bloodstream and recirculate. So in concert, there is vascular integrity that is enhanced by agonists and vascular leakage that is enhanced by antagonism. And the S1P1 tone seems to play a critical role in both these vascular functions as well as in the movement of lymphocytes across the sinus lining barrier. There are 5 high-affinity receptors for sphingosine 1-phosphate shown here on the dendrogram of mammalian GPCRs. These are class A GPCRs in the rhodopsin family. They're called EDGE as in endothelial differentiation genes, as originally described by Tim Hilla and colleagues. And they live in a cluster with the most closely related receptors for the lysophosphatidic acid receptors on this edge of the dendrogram. The closest related receptors are the cannabinoid receptors, which also regulating the tonic events mediated by anandamide have very, very similar mechanisms of regulation. These are high affinity receptors, around 0.5 nanomolar KD for S1P on S1P1, and as I've pointed out, they regulate a broad degree of physiology and pathology. So in order to really understand these relationships between expression, signaling, and function in these complex organ systems, we really took the approach of dissecting it by making tools for the various receptors, both chemical and genetic. And so we made orthosteric ligands, both agonists and neutral antagonists, that occupy the same binding pocket as the natural ligand. We found in the process that we could find or define allosteric ligands, which don't compete with the natural ligand, or bitopic ligands that compete in part for the binding of the natural ligand. We were able to solve, in collaboration with Ray Stevens and colleagues, the high-resolution crystal structure of the S1P1 receptor liganded with an antagonist originally synthesized by Chi-Wei Wong and one of his graduate students in the graduate program. And then we've also made fluorescently tagged receptor knock-in mice to try and understand this. In seeking to look for ways of modulating autoimmune disease in a tolerable way, we hypothesized that both efficacy and safety could be enhanced by having selective, biased agonists of the sphingosine 1-phosphate receptors. And so in 2006, we initiated an allosteric modulator assay development program This went into high throughput screening when there were around 70,000 compounds in the NIH library. The hits were then reconfirmed, selected through dose response and selectivity assays across the 5 high affinity receptors, and then went through mechanism of action confirmation, where we looked for both canonical signaling and non-canonical signaling. In other words, signals that were generated through the G-alpha signaling pathway, G-alpha-I, that modulates S1P1 function, and non-canonical signals that were generated by other parts of the receptor to define bias and to define signals that correlated best with efficacy. Having confirmed that mechanism of action, we looked for antagonist effects because we had a selective competitive antagonist, and we asked whether we could define competitive or non-competitive antagonists and develop a binding pocket hypothesis. Were we binding in an orthosteric, in an allosteric, or bitopic space? Confirming the binding mode by mutagenesis and in parallel developing the crystal structure, we provided direct evidence for orthogonal binding interactions within the receptor that gave us some insights into receptor function. In parallel, we selected for potency, selectivity, stability, structure activity, relationships, and drug metabolism components of the compound, and were able to select a compound, RPC 1063. That was then licensed out to Receptos and and for which an investigational new drug application was filed and it entered man. So how do we know that these agonists are different to simple orthosteric agonists? So here is the NIH compound collection. It's just a diversity plot squashed into 2 axes, so it's B cuts by B cuts. In one corner of the library are a series of hits that are just coded according to potency. The bigger the circle, the larger the potency. Red is S1P1, green is S1P3. The best of these were around 5 nanomolar in EC50 for the receptor, and these were a cluster of aryl oxidiazoles. These ligands induced internalization of the receptor, shown here with a GFP receptor, tagged receptor moving from the cell surface into the lysosomal setting. The receptor is phosphorylated in the third intracellular loop as well as in the C-terminal tail, and one can see ligand-dependent phosphorylation of the receptor, and then variable activation of an E3 ligase that polyubiquitinates the receptor and drives the receptor to lysosomes. And ligands vary. They can be equipotent on the receptor, yet vary by 3 logs in their ability to recruit an E3 ligase and drive the receptor to degradation. And that, we think, truly is the basis of bias. In looking at the receptor and looking at the points of the receptor that made critical interactions with the ligands, we found that the chemical biology of the screening was actually somewhat predictable because there are gatekeeper residues, shown here at leucine at 276 in S1P1, that opens up the binding pocket. It's replaced in S1P3 by a phenylalanine at 263. This impacts on the shape of the binding pocket, and the result is that the compounds that you get in an S1P1 versus an S1P3 screen are almost entirely non-overlapping, despite the fact that the natural ligand, of course, is shared. So shown here is this 3-log shift. Sphingosine 1-phosphate is a very poor polyubiquitinator of the receptor and largely drives the receptor to recycling. Analogues of FTY 720 are extraordinarily efficient at polyubiquitinating the receptor and driving it to degradation. And the ligands that we found out of the NIH Compound collection were in fact somewhat intermediate between FTY720 and sphingosine 1-phosphate. So they were able to drive polyubiquitinylation, but were quantitatively halfway between the two classes of molecule. What was striking, though, about these ligands coming out of the NIH collection was that they showed non-competitive antagonism with W146, the orthosteric antagonist. So shown here is the competition for sphingosine 1-phosphate, and it's just right-shifted and can be fully overcome at higher concentrations. In contrast, the activation by CYM5442, one of the analogs of the hit originally discovered in the NIH Compound collection, shows loss of signal in the presence of the antagonist, and the agonist does not outcompete the antagonist if the antagonist sees the receptor first. So it's a true non-competitive agonist. And when we looked at this non-competitive agonist, we were able to define two sorts of mutations within the receptor. The head group binding residues, the glutamate at 121, the arginine at 292, that provide the salt bridges that bind the aminophosphate head group of sphingosine 1-phosphate, when mutated, lead to loss of S1P signaling, but have no effect at all on the activation of the receptor by these atypical agonists. However, deep within the pocket, there is a tryptophan residue at 269. And mutation of that tryptophan residue has no effect. If we mutate it to phenylalanine, tyrosine, or leucine, it has no effect whatsoever on the binding or activation of the receptor by sphingosine 1-phosphate, FTY720, or any of the orthosteric agonists. However, it completely abolished the activation of the receptor and the binding to the receptor of the alternate binding mode agonists like CYM5442, showing that they were making a specific interaction with that aromatic, with the indole ring of the tryptophan that turned out to be quite important. Shown here is the competition binding data. When you compare the wild-type receptor to the tryptophan 269 leucine mutation, S1P binds perfectly normally. The orthosteric antagonist W146 binds perfectly normally, and CYM5442 is no longer able to displace sphingosine 1-phosphate from the receptor. So we now know that the receptor has at least three parts to it. It's got within the binding pocket, and shown here for S1P3, where we've been able to prove this as well very easily from a biochemical perspective, there is the orthosteric pocket. There is an allosteric set of interactions that are adjacent to the orthosteric pocket but are separable. And then there are ligands that are bitopic and can compete for both orthosteric interactions as well as the allosteric interactions. And it is often the bitopic molecules that are particularly interesting in terms of function and physiology. So where is tryptophan 269 and why does it matter? The interaction with the tryptophan 269 turned out to be absolutely critical because it removed us from having a need for head groups. It altered the physical properties so that we emerged very easily with oral bioavailability, an extraordinarily low maximal concentration in plasma, and an excellent brain-plasma ratio. All of these things were the fundamental pharmacokinetic underpinnings of good tolerability. So to understand this better, let's take a look at the structure of S1P1. Just to remind you, a slide that I got from Ray. Stephen's using his enophiles analogy for receptor binding, where the A2A receptor, the adrenergic receptor is the beta-2 adrenergic receptor is the claret glass. The A2A receptor is the champagne flute. The CXCR4 receptor is the Chardonnay glass. And to my disgust, as I really do dislike martinis, is it turned out that the pocket of the S1P1 receptor is in fact a martini glass. And please pay attention to this key structural element, which is the 3 olives on a stick. And if one looks at the binding pockets, one can see that the diversity, even though they have very similar backbones, the helix tilts and the residues within the pockets make the cavities very different in terms of size and shape and electrostatics. Shown here from the very occluded binding pockets for rhodopsin to the rather more expansive chemokine receptor to the adrenergic dopamine receptors and so on. So what does S1P1 really look like? So the first is the 3 olives on a stick are represented here as a very tight N-terminal alpha helix. We know that this is asparagine 16 because we couldn't cleave the N-acetyl-glucosamine, and so we were left with 1 glucnac on the N-glycosylation site in the alpha helix. And closely allied to it are the extracellular loops 2 and 3, especially with their intra-loop disulfide that keeps a hairpin bend and provides a very tight structure. So that if one looks at the receptor on fast, looking from the outside in, one sees that the binding pocket is completely occluded in the crystal structure when looking from the outside by the N-terminal alpha helix and then the extracellular loops. The only obvious access to the binding pocket is in the large gap between TM7 and transmembrane domain 1, where you can see the antagonist ligand W146, which is required for stabilization of the crystal structure peeking through, and that this gap, in fact, is below the limits of the lipid bilayer and reflects a probable diffusion event within the lipid face that is required for the entry of the ligand. So it's proposed that the ligand binds to or inserts in plasma membrane and then travels horizontally within the lipid bilayer to pick up its interactions with the receptor and sort of like a like one of those little 6-legged bugs with the sticky legs that you can throw at the window and always have 3-point contact as they move down the binding pocket until they reach the floor. It does exactly that until it reaches the binding pocket and is stabilized at floor level. And in keeping with this is the relatively slow on rate. S1P1 doesn't saturate in 2 minutes like a peptide receptor does, but it requires somewhere between 8 and 12 minutes in order to saturate, probably reflecting the slow on rate. But once stabilized, of course, the off rate is glacial. So if one looks at the interaction of the antagonist here in the orthosteric pocket, one can see that the tryptophan at 269 forms a base on the pocket and is just below the binding position of the ligand. We had expected, or many people had said to us, that acyl chains really are greasy, have very little structure, probably just glob on to itself in a rather disorganized way, and that you are unlikely to see an extended structure in the pocket. However, that proved not to be the case, and there were specific hydrophobic interactions that led to a very extended acyl chain, and we could actually visualize all of the carbon backbone atoms of the acyl chain deep within the pocket. And just to this side of the tryptophan at 269. And then, of course, there are the electrostatic interactions with the head group that keep the aminophosphonate, in this case, anchored. And so the binding energy comes from the head group, which gives about 3 logs of potency, and then the hydrophobic interactions that contribute another 6 logs of potency to the binding interactions. So how is the receptor triggered? It's clearly a volume-triggered receptor. The evidence for this is simple. The antagonist W146 has a 6-carbon acyl chain. Extending that acyl chain to 9 or 10 carbon units switches the antagonist into an agonist. If you try to dock the ligand into the receptor using the 2.8 angstrom resolution crystal structure, what one finds is that the 6-carbon chain fits in perfectly, and the ligand binds, makes both the head group interactions and the specific hydrophobic interactions seen in the crystal structure, and all is well. However, as you extend that carbon chain, you can make the head group interactions, but not the hydrophobic interactions. Or you can make the hydrophobic interactions and not the head group interactions, and you lose binding potency unless one small change is made in the binding pocket. And that is to flip tryptophan 269 from cis to trans. And as you flip that from cis to trans, you open up the pocket and you're able to now accommodate the agonist ligand binding both to the hydrophobic sites within the pocket and making a productive head group interaction. So that cis-trans, that rotamer switch probably plays an important role in the modulation of the anatomy of the pocket to accommodate different ligands. So have these altered binding interactions that explore space outside of the orthosphere translated to advantages in clinical pharmacology? So I pointed out that one gets very different intrinsic properties from molecules that don't have a head group. So they have advantageous pharmaceutical properties, they have selectivity, they have picomolar potency, and are really quite clean. And the hypothesis was that these should have potential for an improved cardiac profile, both limited bradycardia and no effect on the QT, the corrected QT interval, no effects on blood pressure. We hoped that they might have reduced hepatotoxicity, and we hoped that with short pharmacokinetics of single daily dosing, that we could actually have molecules where lymphocyte recovery could be entrained on the removal of drug, and 5 half-lives later, 5 days later, patients are essentially back to normal, which may be important in the face of an intercurrent infection in patients who are immunosuppressed. So, just to remind you, what is 1063? It's a selective, well-tolerated oral treatment, we hope, for serious autoimmune diseases that came from the Roberts and Rosen labs at TSRI. It's been effective single daily dosed for relapsing remitting MS with 6-month tolerability that is comparable to placebo. It's been a long odyssey because the first description, as Ben mentioned, was in 2002 from my group. And this is the first new chemical entity that began as a hit from the NIH Compound collection and from the Scripps Screening Center. What I'm going to show you is that disease-modifying treatment works because it provides multi-step interdiction of disease process that inhibits acute attacks and prevents long-term tissue injury. So the screen was run in July of 2006. 1063 was synthesized and fully characterized in May of 2008. The patent was filed that same week. In April of 2009, Receptos was founded, and the preclinical studies for 1063 and the GLP program was initiated. By the end of 2010, it was ready for first entry into man. It was sort of an interesting time for those people that have dealt with spinning out IP from TSRI and starting small companies, in that we had the compound in 2008, and of course Lehman Brothers collapsed 3 months later, making the capital environment really quite different and difficult. But if you stick to your guns, you can find a way to push IP forward. And now we have compounds that are in phase 3, right? 1063 is in phase 3 for 2 indications in a well-capitalized company that is now a mid-cap company. So why does this matter to TSRI? Well, it matters to TSRI because there is intellectual property composition of matter patents that were filed in 2008 that will provide a royalty stream to TSRI on any compounds that are in the clinic and selling in the range sort of from 2018 to the end of 2033. And this has the potential to change the flexibility that TSRI has in approaching the future. So RADIANCE was the two-year MS phase 3, and that is currently running and will be done in 2017. Sunbeam is the phase 3 1-year study that's going to run in that's running in parallel. Touchstone was the ulcerative colitis study, the early data of which emerged about 2.5 or 3 weeks ago. And it's now gone into phase 3 as well as a phase 2 in Crohn's disease, because this was the first evidence for clinical efficacy in ulcerative colitis. So RADIANCE was a phase 2 study for examining the effects of this compound in multiple sclerosis, and it was a screening period followed by randomization, a 6-month treatment arm or placebo, and at the end of that 6-month arm, you have a blinded safety extension where all patients, if the compound was found to be safe, would be randomized to active and would continue receiving active compound to provide safety data. The endpoints were the appearance of gadolinium-enhancing lesions and their resolution as measured by magnetic resonance imaging, and then the calculation of an annualized response rate. What was striking was that of the patients that went on treatment, and the 2 doses were 0.5 milligram and 1 milligram because this is a low picomolar compound, greater than 98% of patients who started on drug completed the 6 months and were still on drug. So this was really well tolerated. There was a very high rate of study compliance and completion. And just as a contrast, the discontinuation rates for dimethyl fumarate Tecfidera in its phase 2-3 ENDORSE study range from 14 to 23%. So competitive agents for MS have a high rate of adverse effects and high rates of discontinuation. This is really positive for the TSRI compound. Now, if one looks at the primary endpoint, which is the cumulative number of gadolinium-enhancing lesions from weeks 12 to 24, we see that on placebo, there were around 11.1 lesions in the placebo group. This was reduced to around 1.5 lesions in both of the treatment groups, and there was no difference. So these were highly significant suppression of gadolinium-enhancing lesions. And gadolinium-enhancing lesions reflect leakage of plasma and other components into the central nervous system as a result of inflammatory damage, and suggests that the inflammatory damage is being shut down. The secondary endpoint was the mean number of gadolinium-enhancing lesions, and you can see that there was around a 90% decrease compared to placebo. Again, very, very positive for the patients in that they have something that is well tolerated, that they can continue to take, and it is controlling the disease process quite significantly. The study was too small to look at annualized relapse rate, and it was only a 6-month study, but in the high dose group, the 1 milligram group, it was almost at statistical significance, even though it wasn't powered for that. And of course, the fully powered data will emerge from the 2-year RADIANCE studies. Now, for patients who have an autoimmune disease, multiple sclerosis, that is damaging the white matter of the brain and is also leading to axonal severing and neuronal loss over time, one wants to try and avoid additional insults. So one looks obviously at the serious adverse effects, and for 1063 there were no notable cardiac, pulmonary, ophthalmologic, infectious or malignancy adverse effects observed. A couple of patients had elevated liver enzymes, only ALT and not AST at all, and all of these resolved entirely on continued therapy, so they were clearly not compound related. The cardiac safety profile was really clean, was much the same as a placebo, and if one looks at the Holter study, looking at effects on QT, for instance, in a thorough QT study, there is none. And if one looks at bradycardia and compares it to the 8 beat per minute drop seen with Gilenya FTY 720 after the first dose, one can see that these compounds are really well tolerated, and through the entire course of treatment did not produce a change in a bradycardia or change in heart rate that was equal to 2 beats per minute. It's always less than or equal to 2 beats per minute, which means it's really quite well tolerated. Now, all of the competing compounds in this area have a degree of hepatotoxicity. 1063 had no significant elevation of transaminases that was drug-related. So, the compound that was discovered here at TSRI and pushed through preclinical development and into man shows good significant reductions for all MRI measures of multiple sclerosis disease activity. The tolerability and safety profiles suggest that there is a good risk-benefit for these patients and that patients will derive significant benefit. And obviously, these data support the rapid development and the rapid enrollment of patients in these trials is a reflection of the interests of both patients and physicians to access this compound for the benefit of their patients. So the key factors that we focused on in the Rosen and Roberts labs, namely the pharmaceutical properties, the selectivity, and the potency, but especially the intrinsic physical properties that allowed us to solve the pharmaceutical properties, really paid off big in that we have a picomolar compound with a very low maximal concentration in plasma that has the tolerability of placebo in 6-month studies, and the phase 3 2-year study started a year ago. So clearly long-term data will be accruing quite rapidly. So why does S1PR1 work so well in disease? I just want to finish by talking a little bit about multi-point interdiction of the immune response. And some of these studies come from our work in a mouse model of multiple sclerosis, extrinsic allergic encephalitis, EAE. But using the S1P1 knock-in that Stuart Cahalan made, the S1PR1 is expressed on neurons, on astrocytes, and endothelia in the brain and in the spinal cord. And it doesn't require full inhibition of lymphocyte recirculation for these compounds to work really well. So the minimalist pharmacokinetic pharmacodynamic relationship for efficacy is around a 30% sequestration of lymphocytes during the dosing period, and you can actually return lymphocyte recirculation to normal within the dosing period and still achieve excellent efficacy. So shown here is the efficacy looking at clinical score, comparing vehicle to a tool, S1P1 selective compound, CYM5442. You can see the infiltration of the optic tract, which is inhibited by CYM5442, as is the demyelination. The lymphopenia, though, that is required to do this is only for 1/3 of the dosing interval. Shown here, lymphopenia peaks at around 6 hours. By 16 hours, in fact, circulating levels of lymphocytes had in fact restored to normal. And the reason for this is that these compounds have a 10 to 15 to 1 brain plasma ratio. And so what happens in the periphery is much less important than what happens within the brain. So persistence of the compounds within the neurons and within the central nervous system leads to down modulation of neuronal and astroglial S1PR1, shown here in these blots. In the presence of agonist, we see polyubiquitinylation of the receptor and degradation of the receptor in these EGFP S1P1 knock-in homozygotes. We see that there is expression on neurons that diminishes on agonist treatment of the neurons, and the same expression on astrocytes being down modulated by agonist treatment of these mice for EAE. So where are the multiple points? I've mentioned so far that it's very important to have impact on lymphocyte recirculation, so the lymphocyte receptor matters. The blood-brain barrier and the endothelial barrier matters. Agonizing S1P1 tightens endothelial endothelial junctions, and you have diminished leakage of plasma components into the interstitial spaces. We've pointed out that, as shown genetically by Gerald Chun, the astrocyte receptor matters, the neuronal receptor also clearly matters. But are there other cells that may play a role in the amplification of responses to immune stimuli? And here it's really been the collaboration with Michael Oldstone, Yoshi Kawaoka, John Taharo, Kevin Walsh, that has driven our understandings of the regulation of cytokine storm. And here I'm going to draw extensively on the work that has been done at the interface of the labs. So a non-selective S1P receptor agonist, AAL-R, it's like FTY720, is phosphorylated, and it inhibits the immunopathology associated with, in this case, influenza infection, a WSN, a model influenza infection in the mouse. And it does so by suppressing a range of cytokines. Shown here is the phosphorylatable prodrug, AAL-R, compared to its stereoisomer ALS that is not phosphorylated. And ALS, of course, has no effect, whereas the phosphorylated intermediate has its receptor-mediated effects. So if we look now at a pandemic strain, this is the 2009 H1N1, one can see that a selective S1P-R1 agonist dosed orally is sufficient to protect from the lethality of this human pathogenic strain in mice. So how does it do it? S1P1 agonism inhibits the influenza-induced cytokine storm. So shown here are the production of cytokines into alveolar lavage fluid in mice infected with the H1N1 2009. And one can see significant interferon alpha type 1 interferon production that is suppressed with agonist, both non-selective and selective S1P1 selective agonists, and then a variety of cytokines and chemokines, including IL-6, are also suppressed by agonism of the S1P1 receptor. We know that the receptor itself has a direct effect on pulmonary endothelial cells, because if you look at chemokine expression in pulmonary endothelial cells in mice following influenza virus infection, one can see again that S1P1 agonism suppresses the production of CCL2, CCL5, CXCL10 as examples by directly suppressing the amplification of cytokines. And so let me end by thanking once again my lab members and collaborators, without whom this work could really not have been done at all, to TSRI for providing an infrastructure and a set of outstanding colleagues that make this integrated science move like nowhere else in the world. And that if we keep our focus on defining mechanism and looking for points of intelligent intervention that impact on disease and on patients, the future of TSRI will hopefully be not only strong, but get brighter and brighter as we work together in the future. Thank you all very much.